Hello, colleagues. I am Diana Samatkyzy. I am researcher in Laboratory of Genomic and Personalized Medicine in National Laboratory Astana. In this poster presentation, we would like to give you information about results after sequencing of the KRAS gene of colorectal cancer samples in Kazakhstan. Today, we have a lot of genetic research in oncology. Currently, many mutations of the KRAS gene have been identified and their mechanisms are being studied for the development of personalized therapy. When KRAS is mutated, it causes distribution of GTP hydrolysis, enhanced nucleotide exchange. After that, KRAS accumulates in an active state leads, therefore, to continuous activation of signaling pathways, which causes promoting tumor cell proliferation. And research in this direction is important for the management of patients with this pathology. As part of the project, we were supplied with oncological tissue samples from 2 clinics, materials of colorectal cancer. Sequencing was performed on 15 samples with colon adenocarcinoma. All patients signed an informed consent, and Sanger sequencing was performed on exons 2 and 3 of the KRAS gene. Sequencing reaction we carried out using the Big Tie Terminator version 3.1 cycle sequencing kit, and we performed on automatically capillary electrophoresis. And we got the result. We identified 7 mutations in the KRAS gene out of 15 samples. The largest number of mutations fell on the missense mutation of exons 2 and 3. You can see this result in the diagram. We also identified deletions and insertions that were not known. These samples were subject for further verification by PCR. Most of them were found to be pathogenic, have a severe course of the disease, and are not treatable with anti-EGFR drugs. In conclusion, I want to say that further analysis of KRAS mutation in larger number of colorectal samples will give possibility to understand the role of KRAS mutations in the development of cancer in Kazakhstani patients, identify genetics profile of mutation, and correctly address the therapy. Thank you for the attention.